Biomolecules NCERT based single sentence questions. Let's start. In experiment chemical analysis to found organic compounds in living organism, the acid to analyze chemical composition is trichloroacetic acid. Trichloroacetic acid. In experiment chemical analysis to found organic compounds in living organism, tissue collected to analyze is a vegetable or a piece of liver. In experiment chemical analysis to found organic compounds in living organism, filtrate is more technically called acid soluble pool. In experiment chemical analysis to found organic compounds in living organism, retentate is more technically called acid insoluble pool. Molecular weight less than 1000 Dalton, biomacromolecule or biomicromolecule. It is biomicromolecule. Molecular weight more than 1000 Dalton, biomacromolecules. Represent cytoplasmic composition. Filtrate. Contain molecule from cytoplasm and organelles. Retentate. Dried living tissue ash is for the separation of inorganic elements. Inorganic elements in ash, calcium and magnesium. Inorganic compounds in ash, sulfates and phosphates. Amino acid, lipid, sugar, nitrogen bases are biomicromolecule. Protein, polysaccharide, nucleic acids are biomacromolecule. Acid soluble fraction contain biomicromolecule. Acid insoluble fraction contain biomacromolecule. Biomicromolecules of acid insoluble fraction. Biomicromolecules of acid insoluble fraction. Lipids. If amino group is and base group are present on the same carbon, it is called. If amino group and base group are present on the same carbon, it is called alpha carbon. Carboxyl group attached to an R group in lipids. Substituents of methane are amino acids. R group can be methyl, ethyl or CH2 with higher number in lipids. 4 substituent group occupy 4 valency positions in amino acids. Number of carbons in palmitic acid 16. Number of carbons in arachidonic acid 20. Hydrogen carboxyl group, amino group and a variable group are present in amino acids. If R group of amino acid with hydrogen, the amino acid is glycine. If R group of amino acid with methyl group, the amino acid is alanine. R group of amino acid with hydroxymethyl, the amino acid is serine. If amino groups are more and in amino acid, it is base. If carboxyl groups are more in amino acid, it is acidic. Example for acidic amino acid, glutamic acid. Example for base amino acid, lysine. Example for neutral amino acid, valine. Example for aromatic amino acid, tyrosine, phenylalanine, and tryptophan. Then one more, histidine. Fatty acid without double bond is saturated. Some lipids have phosphorus and phosphorylated organic compound in them. These are called phospholipids. If fatty acids are with one or more carbon double bond, they are called one or more carbon double bond. If they are called unsaturated. Lecithin is an example for phospholipid of a cell membrane. Oils have dash melting point. Gingerly oil like oils have, hence remain as oil in winter. Oils have lower melting point. Simple lipid glycerol is trihydroxypropane. If one fatty acid esterified with glycerol, it is called monoglyceride. If two fatty acids esterified with glycerol, it is called diglyceride. If three fatty acids esterified with glycerol, it is called triglyceride. Oils have lower melting point. Gingerly oil. Example, gingerly oil. Lipids with phosphorus and phosphorylated organic compounds are called phospholipids. Sugar plus base, nucleoside. Sugar base phosphate, nucleotide. Purines are adenine and guanine. Pyrimidines are thymine, cytosine, uracil. Nucleosides, purines, adenosine, guanosine. Nucleosides, pyrimidines. Nucleosides, pyrimidines. Thymidine, cytidine, uridine. Adenilic acid, guanilic acid are nucleotides. Nucleotides, they are purines. Thymidilic acid, cytidilic acid, uridilic acid are nucleotides and they are pyrimidines. Lipids classified into fats and oils based on melting point. Lipids with lower melting point are called oils. Lipid found in cell membrane. Lipid found in cell membrane are phospholipids, lecithin. Carbon compounds with heterocyclic rings are present in carbon compound with heterocyclic rings are present in nitrogen bases. Example for phospholipids, lecithin. Phosphate group esterified with sugar of nucleoside called nucleotide. Alkaloids, flavonoid, rubber, essential oil, antibiotic, colored pigments, and gum, spices are secondary metabolites. Secondary metabolites. Sugar, amino acid, lipids, nucleic acids are called primary metabolites. Molecular weights ranging between 18 to 800 Daltons approximately called biomicromolecules. Biomicromolecules, or we can say acid soluble pool. Molecular weights ranging between 18 to 800 Daltons. Organic compounds of acid insoluble fraction. Organic compounds of acid insoluble fraction. Proteins, polysaccharides, nucleic acid, and lipids. Cell membranes and other membranes broken into pieces while grinding and form vesicles. Linear chains of amino acid linked by peptide bonds, proteins. Polymer of amino acid is called protein. One type of monomer repeating n number of times. It is called homopolymer. Protein is heteropolymer. Amino acid with human body cannot synthesize are called essential amino acid. Amino acid which human body can synthesize non-essential amino acid. Most abundant protein in animal world, collagen. Ribulose biphosphate carboxylase oxygenase is most abundant protein in the world. Long chain of sugars are called polysaccharides. Homopolymer carbohydrate example cellulose. Storehouse of energy in plant tissues. Starch. Polymer of fructose is inulin. Storehouse of energy in animal tissues. Glycogen. Right end of glycogen chain is non-reducing end. That left end of glycogen chain is right end of glycogen chain is reducing end. Left end of glycogen chain is non-reducing end. Branches in the form of cartoon are seen in glycogen, that is polysaccharide. Helical secondary structures are present in starch. Polysaccharide does not contain complex helices and hence cannot hold iodine. No appearance of blue color, that is cellulose. Starch iodine combination appear in the form of blue color. Plant cell walls are made of cellulose. Glucosamine, N-galactosamine, N-acetyl galactosamine are the examples of amino sugars. Paper made from plant pulp cotton fiber contain cellulose. Complex polysaccharide of exoskeleton of arthropods is chitin. Can hold iodine molecules in the helical portion, starch. Most complex polysaccharides are homopolymers. Building blocks of nucleic acid, nucleotides. Heterocyclic compounds of nucleic acid, nitrogenous bases. Monosaccharide of nucleic acid, ribose sugar. Phosphoric acid of nucleic acid, phosphate. Skeletal heterocyclic rings of nucleic acid, purines and pyrimidines. Pentose are 2-deoxyribose, that is ribose sugar. Nucleic acid with deoxyribose sugar is DNA. Nucleic acid with ribose sugar, that is RNA. In organic chemistry, structure invariably refers to the molecular formulae. Example, sodium chloride and magnesium chloride. Organic chemistry, two-dimensional view of molecular structure. Example, benzene and naphthalene. Heteropolymers containing strings of amino acids are called proteins. Structure of molecules in the context of inorganic chemistry invariably refer the molecular formulae like sodium chloride and magnesium chloride. Structure of molecule in the context of organic chemistry. Two-dimensional. Two-dimensional view of molecules like benzene and naphthalene. Structure of molecules in the view of physicist. Three-dimensional view. Structure of molecule in the view of biologist. Biologists describe the protein structure biologists describe the protein structure at 4 levels. The sequence of amino acid represent positional information of protein. First amino acid, second, then third, and so on. Linear row of amino acid represent primary structure. If protein is imagined in a line, left end is represented by first amino acid. If protein imagined in a line, right end is represented by last amino acid. If in a protein, first amino acid is also called, in a protein, the first amino acid is also called N-terminal amino acid. Last amino acid in a protein is called C-terminal amino acid. A protein thread does exist throughout as an extended rigid rod. True or false? True. A protein thread does not exist throughout as an extended rigid rod. True or false? True. A protein thread does exist throughout as an extended rigid rod. That is false. A protein thread does not exist throughout as an extended rigid rod. That is true. Two amino acids bound each other by glycosidic bond. Two amino acids bound each other by glycosidic bond. Wrong. That is peptide bond. Peptide bond is formed by dehydration. True. Bond between two sugar molecules is peptide bond. Bond between two sugar molecules is peptide bond. False. Glycosidic bond is formed by dehydration. True. Protein thread is folded in the form of a helix. Fold of protein thread is similar to revolving staircase. Only some portions of the protein thread are arranged in the form of helix. True. Nucleic acid exhibit a wide variety of secondary structures. True. Total portion of the protein thread are arranged in the form of a helix. False. Majority of metabolic reaction does not occur in isolation but always linked to some other reactions. True. In proteins, only dash helices are observed, right-handed. Other regions of the protein thread are folded into other forms, then it is called secondary structure. If the long protein chain is also folded upon itself like a hollow woolen ball, it gives rise into tertiary structure. If in a polypeptide chain or a protein, amino acids are linked by peptide bond, which is formed when which is formed when the carboxyl proteins arranged in the form of linear string of spheres. Spheres arranged one upon each other in the form of cube or plate are called quaternary structure of protein. The bond formed between the two carbon of two adjacent monosaccharides is glycosidic bond. The bond formed between amino and carboxyl group of two amino acid is peptide bond. The bond between the phosphate and hydroxyl group of sugar is ester bond. Between the bond between the two nitrogenous bases is hydrogen bond. Adult human hemoglobin is an example for quaternary structure of protein. Number of subunits in human globin 4. Name the subunits of human globin. Alpha type, 2 chains are present, and beta type will find 2 chains. The two ester bonds formed between two different nucleotides are called phosphodiester bond. One of the secondary structures exhibited by DNA is the famous Watson-Crick model. The two strands of polynucleotides are that anti-parallel that is run in the opposite direction. Number of hydrogen bonds between adenine and thymine 2. In the guanine and cytosine that is 3. If each strand of DNA appear like staircase. In a DNA, each step of ascent is represented by a pair of bases. In DNA, each step of ascent, the strand turns into 36 degrees. One full turn of the helical strand would involve 10 steps or 10 base pairs. Pitch of DNA is with 34 angstrom. Number of base pairs in each pitch of DNA, 10. The raise per base pair would be 3.4 angstroms. Type of DNA with pitch 34 angstrom and raise per base 3.4 angstrom in, that is in BDNA. What we have learned till now is that living organism, be it a simple bacterial cell, a protozoan, a plant or an animal, contain thousands of organic compounds. Organic compounds are biomolecules which are present in certain concentration in the organisms are expressed or measures as moles or cells or moles per cells or moles per liter. One of the greatest discoveries ever made was the observation of that all these biomolecules have a turnover. This means that they are very constantly being changed into some other biomolecule and also made from some other biomolecule. Turnover. Breaking and making is through chemical reactions dash constantly or inconsistently. Constantly occurring in all living organisms. Change of biomolecule one form to another form through various chemical reactions collectively called metabolism. Removal of carbon dioxide from amino acid, making an amino acid into an amine. Removal of amino group in a nucleotide base. Hydrolysis of glycosidic bond in a disaccharide are the examples for metabolic transformation. Metabolites are converted into each other in a series of linked reactions called metabolic pathways. Linked reactions are called metabolic pathways. Metabolic pathways are similar to the dash in a city, automobile traffic. Metabolic pathways are linear or circular. It may be linear or it may be circular. Metabolic pathways dash each other, that is, there are traffic junctions, crisscross. Flow of metabolites through metabolic pathway has a definite rate and direction like automobile traffic. This metabolite flow is called the dynamic state of body constituents. What is most important, that is that this interlinked metabolic traffic is very smooth and without a single reported mishap for healthy conditions. In metabolic reactions, every chemical reaction is catalyzed reaction. Catalyzed reaction, and there is no any uncatalyzed reaction. Uncatalyzed metabolic conversion in living systems. Carbon dioxide dissolving in water, a physical process, is a catalyzed or uncatalyzed, is a catalyzed reaction. The catalyst which hasten the rate of given metabolic conversion are carbohydrates or proteins or fats, proteins. Metabolic pathways can lead to more complex structure from a simple structure. For example, acetic acid becomes cholesterol. Metabolic pathways can lead to a simpler to complex structure. By looking towards example, we have to say glucose become lactic acid is simpler structure to complex structure. Metabolic pathways which leads to the formation of complex structure form simple structures called biosynthetic pathway or anabolic pathway. Metabolic pathways which leads to the formation of simple structures from complex structures is called catabolic pathway. Degradation of a complex structure is called catabolic pathway. Association of simple structures can be called anabolic pathway. Metabolic pathway which consume energy, that is anabolic pathway. Metabolic pathway which release energy, catabolic pathway. Biosynthetic pathways are also called as anabolic pathway. Assembly of protein from amino acid is anabolic. Assembly of protein from amino acid require energy. Degradation of glucose into lactic acid is catabolic path. Degradation of glucose into lactic acid release energy. Metabolic pathway from glucose to lactic acid which occurs in 10 metabolic steps is called glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bonds. When needed, this dash energy is utilized for biosynthetic, osmotic, and mechanical work that we perform, that is bond energy. The most important form of energy currency in living systems is the bond energy in a chemical called adenosine triphosphate, nothing but ATP. The blood concentration of glucose in a normal, healthy individual is 4.2 millimoles per liter to 6.1 millimoles per liter. Hormones of human are measured as nanograms per liter. The most important fact that biological system is that all living organisms exist in a steady state characterized by concentration of each of these biomolecules, rate of turnover of molecule through a metabolic pathway is called metabolic flux. Dash are in a metabolic flux. Biomolecules are in a metabolic flux. Any chemical or physical process moves spontaneously to equilibrium. The steady state is a non-equilibrium state. The steady state in living organisms is a non-equilibrium state. Living process is constant effort to prevent falling into equilibrium. Systems at equilibrium cannot perform work. Systems at equilibrium cannot perform work. Yes, true. Systems at non-equilibrium only perform work. True. Biomolecules of organism is in equilibrium state. False. Metabolism and living state are synonymous. True. Living state may exist without metabolism. False. Without metabolism, there cannot be a living state. True. As living organisms work continuously, they cannot afford to reach equilibrium. Hence, the living state is a non-equilibrium steady state. to be able to perform work. Living process is a constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolism provides a mechanism for the production of energy. Protein which enable glucose transport into cells is GLUT4. Protein which is seen as intercellular ground substance, collagen. Protein trypsin is enzyme. Protein insulin is hormone. Protein which fights against infectious agent, antibody. Protein for sensory reception are called receptors. Percent of water in total cellular mass 70 to 90%. Percent of proteins in total cellular mass 10 to 15%. Percentage of carbohydrates in total cellular mass 3%. Lipids, percentage of lipids in total cellular mass 2%. Percentage of nucleic acids in total cellular mass 5 to 7%. Percentage of ions in total cellular mass 1%. Carotenoids and anthocyanins are pigments. Example for alkaloids, morphine, codeine. Monoterpenes and diterpenes are terpenoids. Example for essential oil, lemongrass oil. Abrin is toxin. Concavalin A is lectin. Ricin is toxin. Vinblastin is a drug. Example for lectin, concavalin A. Curcumin is a drug. Rubber, gums and cellulose are polymeric substances. Formula of sodium, formula of potassium, formula of calcium, formula of magnesium, formula of water. Sodium chloride, calcium carbonate, sulfates and phosphates are compounds. Dash percent in the weight of earth crust, hydrogen 0.14. Percentage of weight in the human body, hydrogen. Human body, hydrogen composition 0.5. Percentage in the weight of earth crust, carbon 0.03. In the human body, carbon 18.5. In the weight of earth crust, oxygen in the earth crust 46.6. In the human body, oxygen 65. In the percentage in the weight of earth crust, nitrogen very little in earth crust. In human body, nitrogen is 3.3%. Percentage of weight of earth crust, sulfur 0.03. In human body, sulfur is about 0.3. Sodium in the earth crust, earth crust 2.8. Percent weight in the human body, sodium is 0.2. Calcium, earth crust, 3.6. In the human body, calcium in the human body 1.5. Earth crust, magnesium. Earth crust, magnesium 2.1%. In human body, it is about 0.1%. Silicon. In the earth crust, silicon 27.7%. In the human body, the silicon is negligible. Proteins with catalytic power are named as enzymes. Almost all enzymes are proteins. Nucleic acid which behave like enzymes is ribozyme. One can depict an enzyme by a line diagram. An enzyme, like any other protein, has a primary structure, which means amino acid sequence of protein. An enzyme, like any protein, has the secondary structure or tertiary structure. Has both secondary structure as well as tertiary structure. When you look at the tertiary structure, you will notice that the backbone of the protein chain fold upon itself. The chain crisscrosses itself, hence many crevices and packets are made. They are called as. One such packet is called active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. The enzymes through their active site catalyze reactions at a high rate. Catalysts work efficiently in high temperature are inorganic catalyst. Catalysts work efficiently at high pressures are inorganic catalyst. Catalysts which are damaged at high temperature, say about 40 degrees centigrade, are enzymes, organic catalysts. Enzyme isolated from organisms who normally live under extremely high temperature like hot winds and sulfur springs were stable and retain their catalytic power even at high temperatures like 18, 90 degrees centigrade. Yes, true. Thermal stability is an important quality of enzymes which are isolated from thermophilic organisms. Change of chemical compound is simply refers to a change in shape without breaking bonds is a physical process. Change in a state of matter is a chemical reaction. When bonds are broken, the new bonds are formed during transformation. This will be called a chemical reaction. Ice melts into water is a physical process. So BOH2 plus H2SO4 that form BaSO4, barium sulfate and water, a given reaction is, it's an inorganic chemical reaction. Water becomes vapor is physical reaction. Hydrolysis of starch into glucose is an organic chemical reaction. Rate of physical or chemical processes refers to the amount of product formed per unit time, can be expressed as rate equal to delta P by delta T. If the direction is specified, the rate of physical or chemical processes can also called as velocity. Rate of physical or chemical process refers to the amount of product formed per unit. It can be expressed as rate equal to delta P by delta T. If the direction is specified, the rate of physical or chemical process is also called velocity. Rates of physical and chemical processes are influenced by temperature. A general rule of thumb is that rate doubles by for every 10 degrees temperature. Rise or decrease. Rise in either direction. Uncatalyzed reactions proceed at rates vastly lower. And the general rule of thumb rate decreases by half every 10 degrees centigrade and either direction, that is decrease. Uncatalyzed reaction proceed at rates vastly lower. Carbon dioxide dissolve in the water form carbonic acid. Number of carbonic acid molecule formed in 1 hour in general, that is 200 molecules are formed. By using the enzyme carbonic anhydrase, that is going to be 6 lakh molecules of carbonic acid formed per second. Carbon dioxide dissolves in the water and form carbonic acid. The enzyme carbonic anhydrase accelerated the reaction rate by about 10 million times. The power of enzyme is incredibly indeed. Catalyzed reactions proceed at rates vastly lower or higher. The given reaction is an example for catalyzed reactions have great velocity. The catalyzer in this reaction, carbonic anhydrase enzyme. There are thousands of types of enzymes catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction, when each of the steps is catalyzed by the same enzyme complex or different enzymes, is called a metabolic pathway. Glucose turns into pyruvic acid. That in the given reaction, actually a dash in metabolic pathway, say metabolic pathway, in which the glucose become pyruvic acid through 10 different enzyme catalyzed metabolic reaction. The conversion of the glucose to pyruvic acid in this reaction at a stage, the given metabolic pathway with 1 or 2 additional reactions gives rise to a variety of metabolic end products. That is true. In our skeletal muscle under anaerobic condition, pyruvic acid converts into lactic acid. In yeast during fermentation, pyruvic acid converts into ethanol or ethyl alcohol. Chemical or metabolic conversion refers to a reaction. In chemical or metabolic reactions, the chemical which is converted into a product is called substrate. The protein which are with three-dimensional structures including an active site convert a substrate S into a product are called as enzymes, and symbolically this can be depicted as S P. Substrate S has to bind the enzyme at its active site within a given cleft or packet. The substrate has to diffuse towards the active site. When substrate and enzyme binds each other, they form enzyme substrate complex. This complex formation is transient phenomenon. When substrate is bound to the enzyme at active site, a new structure of the substrate called transition state structure is formed. When substrate is bound to the enzyme at active site, Very soon, expected bond breaking or making is completed. Bond making or breaking is completed. The dash is formed and released from the active site product. Conversion of substrate into the product in the presence of enzyme must pass through transition state structure. Transition state structure. Altered structural states are present between the table and product. All the intermediate structural states which are present between substrate and product are unstable. Stability is something related to dash energy status of the molecule. Energy status of the molecule. In the given diagram, number 1 is y-axis. That is potential energy. Number two is substrate. Number 3, transition state. 4, activation energy without enzyme. Number 5, activation energy with enzyme. Number 6, product. Number 7, x-axis, the progress of reaction. Y-axis of the above graph represent potential energy content. Axes which represent the progression of the structural transformation of states are through transition state, that is x-axis. Energy level difference between S and P. If P is at higher level than the S, the reaction is. Energy level difference S and P. If the P is at lower level than S, the reaction is exothermic reaction. Energy level difference between S and P. If P is at a higher level than the S, the reaction is endothermic reaction. Reaction, one need not supply energy by heating or in order to form the product, that is exothermic reaction. Spontaneous reaction is exothermic reaction. Energy requiring reaction is endothermic reaction. Reaction with the S has to go through much higher energy state or transition state, both. Difference in average energy content of S from that of this transition state is called activation energy. They eventually bring down the activation energy and make transition of S to P is more easy. Enzymes. Each enzyme has a specific binding site, that substrate binding site we say, in its molecule, so that a highly reactive enzyme substrate complex is produced. Dash complex is short-lived. Enzyme substrate complex is short-lived and dissociates into its products P and unchanged with a enzyme with an intermediate formation of enzyme product complex. The formation of enzyme substrate complex is essential for catalysis. E plus S, ES, EP, E plus P, S, ES means enzyme substrate complex. EP, enzyme product complex. E plus S means enzyme ready to bind with substrate. E plus P means product is separated from enzyme. Substrate bind to the active site of the enzyme fitting into the active site. That is the substrate binds to the active site of enzyme fitting into the active site is first step in the catalytic cycle of enzyme action. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. Second step in catalytic cycle of enzyme action. Substrate break the chemical bonds of the substrate and the new enzyme product complex is formed. That is third step in the catalytic cycle of enzyme reaction. Third step in the catalytic cycle of enzyme reaction. Enzyme releases the product of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. That is the fourth step in the catalytic enzymatic reaction, catalytic cycle of enzyme action. Activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of protein. Temperature, pH change in substrate concentration being specific chemical that regulate its activity are the factors which affect enzymatic action. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature is called optimum temperature. Each enzyme shows its highest activity at a particular pH called optimum pH. Activity of temperature and pH decline both below and above the optimum value. Temperature which preserves the enzyme in a temporarily inactive state, low temperature. Temperature which destroys the enzymatic activity, high temperature. Temperature which can denature the enzymatic protein, high temperature. Denature or destroy, doesn't matter. Both are same. That means at a high temperature it happens. With the increase in substrate concentration, velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity which is not exceeded by any further rise in concentration of the substrate shown as Vmax. If the enzyme molecules are fewer than the substrate molecule and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecule. It is shown in the form of Vmax. Activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off enzyme activity, the process is called inhibition. Process is called inhibition. And the chemical is called an inhibitor. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by a 4-digit number. When the inhibitor closely resembles the substrate in its molecular structure, it inhibits the activity of the enzyme. It is known as competitive inhibitor. Enzyme which catalyzes oxidoreduction between the two substrates S and S, oxidoreductases, or dehydrogenases. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Such inhibitor is called competitive inhibitor. Enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and substrate S1, transferases. Enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, carbon-carbon bond, carbon halide bond, phosphorus and nitrogen bonds are hydrolases. It resembles like a substrate that can bind with an enzyme. Consequently, the substrate cannot bind, and as a result, enzyme action declines. Such inhibitor is called competitive inhibitor. Enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bond, liases. Inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure, is the example for competitive inhibitor. Include all enzymes catalyzing interconversion of optical, geometric, or positional isomerase. Isomers, that is isomerases. Enzymes catalyzing the linking together of two compounds. Enzyme which catalyze joining of carbon oxygen CaCNPO ligases. Type of inhibitor which are often used in the control of bacterial pathogen. Competitive inhibitor. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Type of reactions they catalyze. So S reduce, S1 oxidize. S oxidize, S1 reduce. Enzyme used for this reaction is oxidoreductases or dehydrogenases. Oxidoreductase or dehydrogenase. Sg plus S1 results into S plus S1 S plus S1G. Enzyme used in this type of reaction. Transferred. Here, transferases. The type of enzyme work in this reaction is liases. Enzymes are composed of one or several polypeptide chains. True. Enzymes are composed of polysaccharides, pols, non-protein constituents which are bound to the enzyme to make an enzyme catalytically active in many number of cases are called cofactors. Protein portion of enzyme which works with cofactor is called apoenzyme. Three kinds of cofactors are prosthetic group, coenzymes, metal ions. Organic compounds which tightly bound to the apoenzyme are prosthetic groups. Organic compounds with their association with the apoenzyme is only transient, usually occur during the course of catalysis, that is coenzymes. The number of enzymes required for dash for their activity, metal ions, which form coordination bonds with side chains at the active site at the same time from one or more coordination bonds with the substrate. Serves as the cofactors in a number of different enzymes, coenzymes. In peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Prosthetic group, which is a part of the active site of the enzyme, is heme. Essential components of many coenzymes are vitamins. Coenzyme nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate contain the vitamin niacin or nicotinic acid. Cofactor for the proteolytic enzyme carboxypeptidase, zinc. Catalytic activity is the last when the dash is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. That is cofactor. That's it. This is biomolecules, and thank you.